Work. So today we're going to talk about my favorite topic, gene editing. Now, gene editing is certainly something you probably all heard about somewhere. It's really not new on any kind of in pop culture, obviously. And it's hard to even today to look at it and come across something related to gene editing in research perspective. So with it being so prevalent, in our lives, it's a good chance to talk about what exactly it is and how does it work. I know we have a couple of scientists and a biologist in the audience, so it's going to be a little bit of a refresher, but I'm just going to try to make it fun for everybody today. So the definition of gene editing is this: the direct manipulation of an organism's genes using biotechnology or any kind of artificial means. So let's break down this, the hard parts of this definition. Genes. Let's talk about genes. Probably a little bit easier to understand for everybody here. Every organism has, even our friend here, Nicolas Cage. Yes, he is actually human. He is an organism. Nicolas Cage here and all of us are made up of trillions of cells. And each of these cells...
Analogy.
I like Nicolas Cage.
I like it.
And within the cell, in the middle of this, has the nucleus. If you look into the nucleus, it's packed with something called chromosomes. If you take a chromosome and unspool it about, you have long, long strands of what's called DNA. And that's a bug there.
Is DNA of DNA?
Jurassic Park moment.
And
That was CRISPR right there.
A gene is any part of that DNA that codes for a function in the cell and by extension, the organism. So it controls pretty much every aspect of your life, whether it be your sleep cycle, your safety rhythm, the timing or the rate of when your cells divide or don't divide. Your sense of smell, everybody has nice smells going, or just talking about isoamyl acetate today?
That's the one!
Thank you. Your eye color, your sense of taste, everything is controlled by our good old friends, genes. Now let's go to the second part of that definition, biotechnology, or any kind of artificial. Now if we want to expand it, if we want to talk about gene editing as a way of manipulating genes artificially, it's not new. In fact, it is as old as human civilization itself, with things like selective breeding of crops. Right? That is part of biotechnology. We select certain characteristics of crops, whether it be something that's more funky or bitter or more citrusy. Humans have bred to make hops that are more of a certain aspect. There's over 120 varieties of hops, but they're all the same species. They're all hops. But in the more laboratory research sense, a big thing happened in 1974 with the development of what is called a transgenic mouse, which is basically a mouse that has genes outside of its own species. And this was developed first by Beatrix Minz and Rudolf Ganesh back in 1974. So even the idea of genetically modified organisms from mammals and mice is not exactly a new piece of technology. But the major breakthrough happened in 1989 with the development of what is called the knockout mouse by these three gentlemen here, Capechi, Evans, and Smith. They developed a technique and methodology that allows researchers and scientists to target and turn off specific genes. That's really important. Because now, with this development that they made, they were able to, researchers can target a specific gene that they're studying, and then turn it off or knock out that gene to see what happens to that gene or into the system, or what it doesn't do. They completely revolutionized our understanding of genetics and certain genes, and completely revolutionized medicine as we know it. So this is a fundamental that we're going to talk about when we talk about gene editing.
That one doesn't happen.
So we're going to talk about their discovery essentially and their methodology. So let's talk about this. How do we target and turn off specific genes? It's basically this. Hijack the cell's DNA repair machine. Yeah, this doesn't make sense, right? How do we get from DNA, how are we getting from gene editing to DNA cell repair and hijack that? Well, let's go through that. Your body goes through a lot of shit. So does your cells. So does your DNA. DNA is very small. It's robust, but it is fragile. And whether it be from your normal metabolism, the poisons that you drink, I'm talking about alcohol, of course, but I don't so much. UV from the sun is going to damage your DNA, right? And one of the most common types of damage that the DNA goes to is called a DNA break. It says it's the definition. So you have to choose strands of DNA, and it breaks by some kind of, for some reason. It happens quite often. Now, cell doesn't like shit like this happening, obviously. You could have like a key gene that breaks, and then the cell can't function properly. The cell has to die, or worse yet, it kicks up into overdrive, it starts dividing, and you have cancer. Right? So the cell needs ways to repair this kind of DNA. And there are two major ways this happens: either by brute force, repair through brute force, or repair through copying. Now, repair through brute force is basically what it sounds like. You take the break of your DNA, and the cell just matches those two together and fixes that break. And that's a little bit of, it's a brute force method, it's a little bit unrefined. So you end up with like parts of that DNA missing, or stuff added to it. And while may not be ideal, that break is fixed. The other way, which is a little bit more elegant, is this repair through copying. That only happens if there's an intact copy of that same DNA that's damaged. You know, when cells need to divide, they need to make copies of every part of themselves, right? So that when they divide, they have equal amounts of DNA in the cell. So if a cell is in that kind of stage, where there's a copy of that broken DNA, the cell will actually use that as a template to repair itself, and you have a seamless repair with this. It's a little bit hard to visualize, so I like making analogies like this. Imagine before the printing press. Monks like these had to make copies and transcribe books, reams and reams of books, so that they can be distributed like this, right? They would always have to make copies. So, for example, what if the monk here is making copies, and a bird swipes in and tears out a couple of pages at a time?
Asshole!
What an asshole, right? But the deadline's coming up. He needs to make more copies of his books. Got to send over. Well, thankfully, if there are books that he's already made copies of, he can just take those and use it as the template to make more copies. And just keeps making the same copies of the book. None's the wiser. That's what repair through copies involves. On the other hand, what if there are no copies that he can refer to? Well, shit. He's going to have to see those 2 pages that remain, and just repair it to make sure that those 2 pages work together some way. Book's repaired, but it's forever changed. So any kind of copy from this is going to happen. It's going to be changed. That's repaired through brute force, but at least it's repaired. So Evan, Smithies, and Capetti, we're looking at this. So these are smart people out there looking at this. Like, is there a way that we can manipulate this kind of cell pathway, the cell mechanism? And they're looking at this, I just thought, wait a minute, there might be something we can do with this. Can we do something here at the intact copy of the DNA? What if, instead of, if the DNA break happens and the cell is using its intact copy within the system, what if we introduce an artificial DNA sequence that's similar enough to where that damage is, but we've changed this just slightly, that if we trick the cell to use that as the repair template, we can actually insert our own DNA sequence in here. and thus perhaps disrupting that specific gene. Right? Let's go back to the analogy of the monk who's making copies. Making copies. None's the wiser. What if some kind of rogue comes in? This is Harvey Mudd from Star Trek, the original series. He comes in, this rogue here comes in, takes a couple of these copies that he made and writes just slightly, takes a couple of pages and writes like mudless here, writes a couple of different phases, and just scurries away. But then Monk's none the wiser. Crow fucking comes back again and rips out his pages again.
Still an asshole.
Still an asshole. We really got to do something about this crow problem. And he needs to find a copy to repair it. What if instead of the true copies, he picks up one of these fiddles around copies that our friend, our rogue, copy, our rogue, basically does it on. Well, then from there, every single one that he copies as that is template becomes a new, becomes basically a new condition. It's different. That is the fundamental principle behind this breakthrough that they did in 1980. They developed a system where you can introduce artificial DNA sequences into mice cells, and if they're to target specific genes, and if in the event that DNA damage happens to that gene, the cell uses their artificial template as artificial DNA sequence as a template to copy, and that therefore disrupts the cell. That is what the knockout mouse is. One of the most other important things they were able to develop is the mouse. The mouse is a mammalian system, so it's closer to humans. There's a lot of other animals that are used, like flies and C. elegans, like flatworms. But doing it in mammals was the big thing. Being able to set that up. And it's absolutely revolutionary. Like I said, it changed the way we understand genes and how we do biology. And of course, they were awarded the Nobel Prize in 2007 for this discovery. And that is the principle behind what gene editing is and how it works. I do have a little bit of time. I'm glad I have to speed through a little bit of things, but I think it is good time to talk at least briefly talk about the CRISPR-Cas9 system. This is probably something that you've heard about. It is like the newest tool that was first came out and made publicly available about end of 2011, 2012-ish. It was right when I was in grad school too. I'll briefly talk about why this is so important, revolutionary, why everybody uses it. It's essentially a tool. You see, when this breakthrough happened in 1989, and the knockout mouse was developed, and this system became prevalent among researchers, it was still a very difficult system to work with. It would cost money to make those certain mice. It would cost very specific techniques to actually do this, and being able to breed these kinds of mice to make sure that they're purebred, as in the sense of like they have that knockout gene in them, was a very meticulous and time-consuming process. It took a long time. It was difficult, it was expensive, it was time-consuming. One of the major reasons for all of these was because in the process of, we talked about, in order for the knockout to happen, we tricked the cell into using an artificial DNA sequence. But this only happens if there's a DNA break, if the DNA is damaged within the cell. And no matter how much you want to try, sure, it happens, but DNA is a long molecule. Having a break at a specific place at the gene you're targeting is a crapshoot, essentially. It will happen eventually, but you're lucky that'll happen. That'll even happen once in the system. So you gotta just keep what they start transfecting, throwing in these artificial sequences into cells. Make sure, and then just wait to see if these cells actually get this insertion in them. So ever since ever since the knockout mouse was invented, everybody was trying to find a way to safely induce a DNA break, and specifically these two things: being able to target a specific gene, and then being able to cut or damage that gene so that it kickstarts the cell DNA repair machinery, so that the artificial DNA gets inserted into the gene. That's what CRISPR does. At the basic principle of it, CRISPR is only a tool that targets a DNA sequence and cuts it. That's all it does. But it does it with fundamental accuracy, and it is so easy to construct in the lab that even if as an undergrad student with basic molecular biology techniques can do it. You didn't need millions of dollars. You don't need millions of dollars of microscopes and facilities to induce gene editing. To be a little bit more specific, what they call the CRISPR is composed of two major components: the guide RNA and the Cas9 protein. The guide RNA basically is constructed to attach to certain parts of the DNA, and so it acts as just a targeter. It searches through a DNA or the genome you're entering it into, and it attaches itself. And once that happens, the Cas9 protein only works when it finds the guide RNA that's attached. And once these two attach, when guide RNA and Cas9 attach to each other, then the Cas9 protein cuts the DNA. And what happens when you cut the DNA? The cell's repair machinery kicks off. And so it makes it easier for that artificial sequence you're entering to get inserted into that damage. I can't stress this enough. It dramatically made shit so much easier. But at the end of the day, CRISPR is a tool. It's not like a revolutionary method of gene editing. That fundamental principle that was developed in 1989 is exactly the same. But the CRISPR system just made this so much easier. And naturally, the developers of the Cas9 or made it available into the mammalian system. Kabandia and Dunga were awarded equal prizes of the Nobel Prize in Chemistry in 2012. So I hope we've learned a little bit about gene editing, what it is, and how does it work today. Just a little thing. This is a little picture I had during my research time. I studied brain development in the sense of like how the brain constructs itself. And so all these cells that you see here these are all gene edited with Cas9. I basically entered Cas9 so that there's a structure protein in here to make sure to either get a red tag or a green tag in it, and if those two combine, it becomes a yellow tag. So you can see some of these are just green, some of these are just red, some of these are just yellow because it's a combination of those. So this is just pretty, but these are all gene edited, neurons, brain cells. All right, thank you very much. I have about 10 minutes of Q&A, so please.
Can I just say that you're like a really good presenter?
Thank you.
I think your presenting skill is obviously you've had experience.
Yeah, well, that's why Kyushu University gave me the and it's all taxpayer money, so thank you very much to everybody.
You're welcome. Thank you. That's really good.
Yes.
A little bit. I'm a medical doctor.
Yes.
And I'm interested in the knockout mouse. I didn't do that, but I'm interested in that. And also, this technology can be used to make a designer baby or something like that.
Yes. So
negative aspect of that, I want to know.
That's true. Negative aspects, of course. Since gene editing is made much easier now, and of course, the first time I did this presentation was good coincidence was like at the third Nerd Nite in Kansai that I did this, and it was like 2 weeks after He Jiankey from China reported on that designer baby. So I think it was in 2018 or 2019 or something. And that caused a whole lot of shit across the world, obviously. And that is the thing. Is it easier to make the designs on babies? Yes. I don't think it's a good thing to do, but it needs to be discussed about, I think. And I think there will be people who are more like willing to, take that step. But, there's too many, there needs to be a lot more discussion and like, a global consensus on what to do about this, because it isn't just about the science at that point. It's it's ethics, it's humanitarian, it's an economical aspect as well. So, and there are people who talk about it, even here in Japan as well. But Japan is actually kind of strict in that sense. They're like doing any kind of gene editing stuff towards humans, especially. Well, that being said, not everybody in the world, like nobody in the world is actually saying like, yeah, let's do it. So it's a fun aspect. It's a good. It is a very good thing. I think we'll hear more about it as time goes on, because who knows? It could be somebody who is already out there, right? There's twins in China, allegedly, that are gene edited. So we'll have to see what happens. I mean, that's who we know about. I think there might be more.
I'm also interested in bioethics, but I'm not going to ask like general question. But if you are the one to make these laws, where are you going to set the boundary?
No gene edited babies.
And only that.
Well,
so what are like gene editing adults?
Well, gene editing adults is harder, and like stem cell therapy is already happening, and that's kind of a close thing. But there's a very limited window of what you can do with gene editing adults, right? Because we already have the trillions of cells we have. Unless we are able to edit the trillions of cells as it is, we can't do that. So I think there are cases where there have been treatments of like. Extracting the patient's cell, gene editing them, and then implanting them back in. I think it's a lot of stuff happens with like blood-related stuff, because bone marrow transplantation is a lot easier to do. I think that's fine.
So what about xenotransplant?
What?
Xenotransplant?
Xenotransplant. What's xenotransplant? Well, what is
Like transplanting a pig's heart into human being.
I think that's pretty cool. Yeah, xenotransplant is because we did. That guy just recently died, right? For 2 months with a pig heart.
Oh, died.
Yeah. Yeah, this one right here. Wait. Yeah. I don't know. They probably did use CRISPR for this one, but this is basically what they did was they nuked the patient's immune system, and then gene edited pigs so that they reduced the immune response that organ would produce. And the guy, this was very experimental. The guy was in poor health as it is, and that's why he only survived so much, essentially. But it worked. The guy lived for so long. I think that's fine. It's interesting. I mean, there is more bioethical and economical problems that you have to address with that, right? So that's certainly something worth discussing. But I am certainly for that kind of research. But when it comes to stuff like what they say, editing to the germline, which is basically those edits can be transferred to the next generation. That's a wall for me, I think.
What do you think about like the informed consent? Like if I'm healthy enough, but I just feel like it's cool to get a pig heart transplanted, then I'm volunteer to do that. Is it okay to accompany doing that?
If you can find a doctor who's okay with it and you're okay to do it, then that's fine. You probably won't survive very long. But
So many researchers, medical researchers are interested in the stem cell research of IDS or kind.
Yeah.
So that can combine with the gene editing?
Oh, absolutely. There's already. So I used to work at Kyoto University, and IPS research, and there's a couple of people there. There's a Canadian guy that I really like, Knut, who does specifically. Like his name is Knut Wolder, but he does specifically, how do you apply gene editing to iPS cells? Because it's still very hard to actually gene edit stem cells, because stem cells. This is actually still kind of a mystery. They're very hard to edit, the genes, because they will repair their stuff really quickly. That's one of the reasons why the knockout mouse development is so difficult, because you need to use mice stem cells, and even mice stem cells. are easier to edit than human stem cells, but are still hard to edit. I think it's because since they're so, the pluripotency, which means it can become anything, it really is meticulous at making sure it doesn't fuck up. As it is, right? Because you fuck up at the stem cell stage, whatever happens, that goes down the line to every single part of your body.
So you mean that there's a self-repairing system or something?
That's what I think it is. It's still like hard. People are still looking into that and exactly why, but it seems that the self-repair, it's so meticulous and self-repairing, it has a better repair system at that stage that it's harder to edit.
Something you can never gene edit.
Something you can never gene edit. Like, at least, not at the, like, not off the top of my head. Things are like harder to do because, like I said before, certain cells or certain organisms have better repair machinery to kick off. But at the end of the day, let's just go back to like the CRISPR system. That's only 20 base pairs long, guide RNA. So, like, if that enters the cell and into the DNA, and Cas9 and like this system survives, like you probably could edit it. Maybe even just do, like go through brute force methods or something. But I would be interested to see like if there was one that's like you can't edit. Like, whoa, what the fuck? What do you mean? Like, that's so cool.
It's out of a bottle.
So a little bit.
そのクリスパーシステムのそのアキュラシーを知りたいです。だからそのターゲットをカットするのはどれぐらいその正確 100% or 75%?
That's a good question. で、日本語でちょっと。はい。Let's see. Fengzheng Fengzheng is one of the other people who were in one of the researchers that was in line with possibly getting the Nobel Prize for developing the Cas9 system. His lab developed an algorithm that can perhaps like give you a relative accuracy of how well this guide RNA, because basically how well a guide RNA can attach to the DNA is what's going to determine if Cas9 works. So, and since it's only 20 base pairs, it's easy to like construct, but it's only 20. A lot of DNA is long, so it could attach to somewhere completely like on the other end, and you don't want that to happen. Right? So, like you need to construct the guide RNA to make sure that it's unique enough so it doesn't attach to anywhere else to make a cut, or like a lot of times, so GC rich areas, body and cytosine rich areas are also slightly harder because they don't attach because they require 3 hydrogen bonds to bind up. That causes a little bit of problem, so it's least likely to attach. But Fengzhen's lab actually developed a tool where you can like punch in your code and the organism that you want to use it in. It'll give you like a relative accuracy on how well it will attach. And then the other downstream ones that it could actually attach to. So, 100%難しい。ごめん、日本語で話そうと思って。
It's actually really cool.
Just got really excited.
So, is it easy to making a DNA or RNA sequence artificially?
Yeah.
Is it established technology?
The CRISPR systems are as well established. I mean, so this is 20 base pair, and this is an extra like 15 or 20. So it's like 40 base pairs long. I don't know. 業者で外注した2日で行くんですか
Yeah, of course.
Okay, that's my time.